Okay, next speaker, Lynn McDonald from CSIRO Land and Water in the Carbon and Nutrient Cycling Research Group based at Adelaide, and she's currently leading the South Australian component of the National Soil Carbon Research Program, also quantifying carbon, nitrogen and phosphorus content of soil organic matter, and researching biochar on soil biology. And she's going to be talking to us about soil sampling strategies for carbon.
Excellent, thank you. So yeah, just a little bit of an outline of what I'm going to go through today. We're going to take a little bit of background just in terms of what we're dealing with to sample. Then I'm going to run through a bunch of different strategies for sampling paddocks. So we're going to look at the 25-metre grid. We're going to look at paddock scale random sampling and paddock scale grid sampling, and also what we might do if we need to think about zone sampling of paddocks. I'm then going to finish up with a little introduction to the National Soil Carbon Research Programme with particular kind of focus and highlights from what's going on in South Australia. So just to start off with what we're dealing with, we know that soil forms a continuum in space. It's not a defined unit. We know that it has vertical dimension, so we need to consider depth. We know that there are diagnostic horizons that are not visible from above ground. And we know that soil can be difficult to classify. We know that we're dealing with a variable landscape in terms of soil type and landscape features such as slope and undulation. We know that it changes in terms of texture and colour and mineral composition. And management history, of course, has a fair bit of a large impact on soil carbon and on variability. And I guess just trying to visualise this, I went into Google Maps and just took a little look at the satellite images and grabbed various shots from across the Australian landscape. And I guess here I just want to get across the point that we can visualise this variability in the landscape to a certain extent. And I guess maybe just highlight that out here in a snapshot of WA, we have some pretty big regular paddocks, and they are across a fairly flat kind of sand plain landscape. And that contrasts a fair bit with the type of remnant dune type systems that we get in a lot of other Australian agricultural systems. So we need to consider what we're sampling. Our aim is to collect a representative sample of whatever we have decided. But in order to define a representative sample, we need to carefully consider what our purpose is and what our spatial context is. So just taking a little bit of a closer look at purpose, are we looking to determine change in soil carbon content over time? Well, to me that's not nearly specific enough. It's way too fuzzy around the edges and doesn't really define what we need to know in order to sample correctly. We need to define the spatial area that we're looking at, and we also need to look to see if there's any temporal requirements. So if we're looking at change over time, is there a particular sampling gap between sampling that we need in order to measure a minimal change in carbon content? So I guess looking to better define that question, we might expand the question, or the focus is to determine change in carbon content of a specific paddock after however many years of management change. And then we're beginning to get to the nitty-gritty in order to help us design our sampling strategies. So the sort of minimum requirements that we need are to collect a representative soil sample or samples. We're looking to sample to a minimum of 30 centimetres depth. Bill mentioned that we need an accurate estimate of bulk density and also an accurate measure of soil organic carbon content. We also need to think about the confidence or uncertainty associated with our results, and I guess that's why up here we need to think about the number of samples that we're required to take and how that influences our confidence in the results. So 30 centimetres depth, why? I guess we know that the majority of soil carbon is in the top 30 centimetres, and most of the management management changes impact that top 30 centimetres. There are, of course, always exceptions to the rule. So if we're specifically looking at engineering soils at depth, so clay spreading or delving type approaches, we might need to consider a depth greater than 30 centimetres. And particularly if you're changing to a crop species that has deeper rooting depth, then you might want to consider going beyond 30 centimetre depth because there'll be carbon inputs at the lower zone. But again, that is helping to define, or it's key to define the purpose and the context before defining the sampling strategy. And I guess the other point on this slide is that soil type has an important influence on soil carbon content. So here we've just got a bunch of common Australian soil types, and up above is the change in carbon content with depth. Moving on to bulk density, simply the weight of soil in a given volume. And we need that in order to calculate total organic carbon content on a unit area. So we know that bulk density generally increases with depth, and bulk densities above 1.6 can restrict root growth. But important to our carbon calculations, we need to multiply the depth of our sampling, multiplied by the bulk density, and multiplied by the carbon content. So if we have a 30 centimetre depth, and a bulk density of 1 and a carbon content of 1, then we have 30 tonnes of carbon per hectare. The complication with bulk density comes when we step into gravel soils, and we'll need to apply a correction factor to the calculation in order to account for the gravel content, and sampling soils greater than 10% gravel becomes difficult at more than about 10%. And I think there's one or two people in the audience that have particular experience with sampling bulk densities in difficult soils. So, moving on to our sampling strategies, I think most people will probably be or have heard of the 25-metre grid sampling approach, where a 25-metre grid at 5-metre intersects is put down within a paddock, and it can be laid out relatively quickly. It's a common approach, and it tends to minimise the influence of landscape variability on the results. And this is the approach that's been taken in the National Soil Carbon Research Programme. So just to run through that, we've selected a random GPS location, which defines the bottom corner of the 25-metre grid sampling. We've got 5-metre intersects, so 36 possible sampling points across that grid. At 3 of those, we take a bulk density measurement at 3 depths, so the 0 to 10, 10 to 20, 20 to 30. And we create a composite sample across that from a collection of 10 samples, which are bulked at the 3 different depths again. So that's the approach that's been taken within the National Carbon Project. The advantages of this grid type approach is that we can go back and revisit the precise locations. It's good for monitoring purposes, so we can go back to that landscape. We've minimised the landscape variability, and it provides a good data set where we have high replication or high representation. So for example, in the South Australian National Project, our aim is to determine the impact of management in red brown earth soil types on the carbon content. So we have over 200 sites on red brown earth soil types, so our representation is high. We have a stratified design, so we're specifically sticking to red brown earth soil types. The small size of the sampling grid ensures that we don't cross too many soil boundaries, and we've got this soil times land use combination, which is replicated. However, if we're thinking about sampling at a paddock scale, and our specific purpose is to define the carbon content of a specific paddock, then we want to encompass that paddock scale variability. And if we plop down the 25-metre grid sample within a great big paddock, we're actually losing that paddock variability. So we might like to think about an alternative strategy to get a better sampling or better estimate of the carbon content within that paddock. So an alternative approach would be to randomly scatter a number of sampling points across the paddock and collect samples from there. If you're looking to gain more information about the variability within that paddock, you might like to keep those samples separate. But if you're looking to form an average carbon content, then you might like to composite the sample across those sampling points. I guess when you go out into a large landscape, it can be a little difficult to randomly allocate sampling points and to be happy with your random allocation. You might by chance get clustering in particular areas. So to avoid that, you could put over a larger sort of grid scale or grid across the paddock and then select the specific grid intersects that you want to sample across. And the advantage of that would be it's easier to keep track of where you have sampled and to distribute the number of samples across the paddock in a more uniform manner. So I guess the example that's up there is an image that's grabbed from the kind of sand plains in WA. And again, we've got this probably fairly uniform paddock. And I guess that in some ways is fine, but if we have any sort of underlying variability in our soil across that paddock, then we might run into problems. So just to kind of highlight, if the blue zone represents a different soil type, so if we've got contrasting soil types within that paddock, and we've randomly thrown out our points of collection for our sampling, it might happen that we only have a couple of those samples falling within one soil type, and then we run into potential issues of misrepresenting the carbon content of that paddock. Particularly if you go back in subsequent years and your random sampling falls out at different points, you might on that return trip find that you have more sampling within the alternative soil type as opposed to your first sampling. So the confidence of your results in that case could be lessened. So if there is knowledge about underlying variability in your paddock, you might like to take that into account when you're designing your sampling strategy. So it might be appropriate to redistribute some of those sampling points so that you have better representation of the two different soil types on an area basis. And I guess just a second example there on remnant dune type systems. It's very obvious in this half of the picture that the hills of that dune system are managed quite differently. And that if you're defining your sampling based on a management strategy, then obviously that shrubby type vegetation on the top would be inconsistent with the rest of the paddock and therefore wouldn't be sampled. It's less obvious on this side of the paddock where you've still obviously the dune system going on and you have variability in your landscape characteristics and potentially your carbon content. And it might be important to take that type of variability into account when you're designing your sampling strategy. And in that specific case, you might like to take a similar approach to the underlying variability in soil type and actually provide a zone stratification type approach to ensure that you're incorporating both those hill slopes and swales in the sampling strategy. So in summary, quickly, the minimum requirements are to provide a representative soil sample to a minimum of 30 centimetre depth and will require an accurate bulk density. In order to look at confidence or uncertainty, we'll also need a number of different replicates samples in order to calculate that. It's important to carefully consider the purpose. So is the purpose of the soil sampling to look at change over time? Is it to compare two different sites? Or is it to improve particular understanding of variability? Again, it's important to consider variability. So what background information is known that can be used to inform information on variability? And if there is a change in management practice, will variability change with that management through the two different sampling periods? And I guess I just wanted to highlight that the ISO standards for soil sampling for carbon accounting purposes emphasize that sampling strategy is driven by purpose, but and that the general rules that are applied have to be adapted to that specific purpose and the specific situation. So it is a case of kind of adapting general guidelines, and there isn't specifically one sampling approach that's going to suit all situations. So just to highlight some of the SCARP work that's going on within the national project. The larger objectives of the project are to define a nationally consistent method for quantifying soil carbon. This phase of the project had a lot of discussion before we got to the sampling design across the project. And then beyond that, we're looking to identify in regionally specific areas land management strategies with the potential to build soil carbon. And then, as John's talked about before, there's the development of cost-effective methods for measuring or quantifying carbon stocks. So one component of that is the MIR predictive approach, and the other is the use of automated devices for bulk density, which might make life easier in some cases. The data, of course, is flowing into the national accounting system to improve modelling systems there. So the sampling approach we've run through already, and it's almost consistent across the programme. However, there are always exceptions, and I guess that just highlights the need to adapt the methodology to specific context. So in the rangelands projects, where they're dealing with very large spatial scales and very large paddocks, their grid sampling is a lot larger than 25 metres. I think they're working with a 100-metre grid sample. And Diane Allen's written a nice review of sampling designs specific to the Greys context. So in South Australia, what have we been up to? We are aiming to identify management practices with the potential to change soil carbon content. We've got a relatively small project, so we've only got 200 sites, and we decided to focus on red-brown earth soil types. And we have two target areas, the Mid-North and the Eyre Peninsula, and they are fully sampled at the moment. We decided to break down any sort of artificial boundaries around our kind of land management systems. So we've included everything from permanent pasture to continuous cropping and all the sort of mixed rotational systems in between. So we're treating our sampling set in more of a regression approach, where we're looking at the intensity of agriculture within the system. We've taken a random sampling approach. We have no knowledge of management before we go out there, so we're very reliant on the landowners providing or returning the land management questionnaire. And I know that I'm going to have a pretty unbalanced statistical design. So within the project, we are going to have three major data sets. We're going to have the management histories, which I'll talk a little bit about. We are pulling climatic and topographic data based on the GPS coordinates from the silo data type databases. And we'll have our soil organic carbon content and the fractions that John's talked about before, so particulate organic carbon, humus, and the resistant components. And in order to understand what's influencing soil carbon content, we'll be looking at correlating the management histories and the topographic data to those carbon contents. We know that that soil carbon content is influenced over many years, so we've collected the management data. And the type of information that we've collected, oh dear, is, can I remember, a bunch of information on crop type, tillage management, stubble management, and various aspects of pasture management, so grazing aspects and the likes. And that database is fairly large, and it has provided one or two headaches, but we have got there, and it's a very valuable data set in order to understand our soil carbon. It also fits into probably filling some of the gaps that Bill was talking about in terms of need to improve our understanding of what's going on there in the landscape. So this is nice, up-to-date information, hopefully. And the type of things that we can do with that data is look to see, because we've gone out there in a random manner into our landscape, we can make comment on the proportion of crop systems or mixed systems within our red-brown earth soil types in the target regions. We can look at the sort of frequency of incorporation of legume rotations within the management data in the last 10 years, or the frequency or adoption of no-till type practices. And then we can define our questions in terms of, we might like to ask how many pasture years are required to significantly alter soil carbon content. And that final little schematic is a look at the tillage management or trends in tillage management over the last 10 years. And we can see the adoption of no-till and reduction in conventional till and minimal till across the region. So just where we're at with this at the moment, our field sampling campaigns are complete. We have 130 sampled in the mid-north and 70 sampled on the Eyre Peninsula, 70 sites that is. We've got over 900 samples which are going through various pipelines. In terms of the carbon analysis, our MIR spectra we have across the whole data set. Our fractionation is a good way underway, and that will be used to update the prediction algorithm to get a red-brown earth specific prediction algorithm for these different fractions. One big headache within the project has been our carbonate soils, because 41% of those samples are carbonate, and in terms of analysing samples for carbon, that requires a bunch of pre-treatment with acid in order to get rid of the carbonate and measure the organic carbon content. So that's been a lot more time consuming than we thought. And I guess that just highlights the data sets that we're going to have in terms of the management, the environmental and the carbon data sets. So from there, we're looking to understand the variability in our sample population, first of all purely with the management questionnaire, and secondly looking to identify management drivers in soil organic carbon content over these two regions. In terms of looking at uncertainty, we do have a bunch of spatial sampling. So we've got some samples where we have the grid, the 25-metre grid, and instead of compositing the samples, we have the 10 samples kept separate, and we also have Paddock scale sampling at those sites, where we've scattered 10 sites randomly across the paddock, and we're looking to assess the uncertainty associated with predicting measuring the carbon content in these paddocks based on the 25-metre grid and the paddock scale sampling. I guess just to finish, across I think this was largely the mid-north, this is carbon contents from last year's data. We're looking at carbon content varying from 22 to 64 tonnes of carbon per hectare. And the next stages of our course are to update that with the fractions, to look at the distribution of the organic matter fractions, and also to correlate that data to the management and environmental data to look at what's driving soil carbon content. So just some acknowledgements, firstly to the participating South Australian farmers, because they have been very good at getting involved and getting back the management information. We've had a sampling crew of over 10 strong out there, really putting in the hard work. And the lab crew have been pretty much up to their eyeballs and working pretty hard over the last while. So, thank you.
Lynn, very interesting talk. Can you tell me, I'm interested in your estimates of bulk density, because if they're done using a drilling rig with a coring device, my experience in calibrating neutron probes is that it's very, very difficult to get a reliable estimate of bulk density from a core sample, from a simple drilling rig. Can you comment on how you...
So South Australia, for the bulk densities, we're actually hand coring.
Oh, good.
Yeah. I think, we recognise that there's likely to be compaction with the drill rig. Yeah. And our soils aren't actually within the part of the project that's looking to looking at the bulk density, the rapid bulk density measures. So Dan Murphy in WA's done a fair bit of work with that on the sands out there. And I think there's been other work. Update me, John. Yes. But I'm not sure what region he's been concentrating in or soil types. So, but yeah.
It's really important to get a good estimate of the density.
Yeah, and it's one of the parts of the sampling that takes the longest. So it'll take as long as getting the composite sample together. So,
Lynn, just a sort of a statistical view. How big is the difference that you want to estimate in changes in soil carbon? Because the accounting seems to take it down to tonnes per a 1 tonne difference. And what probability do you want to have in detecting that difference? Because that really determines your sampling number.
Yeah, that is going to determine sampling number completely.
So the
I wouldn't
is
be.
Is it an impossible task to get to a soil
1
assessment
tonne
based on a carbon accounting number?
I think the carbon accounting will have to define what the minimum limit of change is. And that will help define how many samples need to come out the ground in order to provide the type of robust data. Yeah. But at this stage, I would not like to take a gamble on what might be our minimal detection limit.
You mentioned that in order to get an assessment and an appreciation of the uncertainty, that you could analyse the 10 cores individually and see the spread of results. Did you also consider going back to some of the other sites of the 36 pattern grid and take another 10 samples and just look at the variability using that approach?
Sorry, which 36 sites?
When you
the
drew
other
the
grid.
5x5 grid and you took 10, I think I understood that you took 10 of those 36 sites. Did you ever also go back and take another 10 different grid points?
No, we haven't done that, but that would be interesting. Yeah.
So just following on from both of those questions, in respect to the last one, are you bulking those 10 samples before you do the analysis, or are you doing 10 separate analyses and then calculating a mean?
That'll be done on 10 separate, they've been deliberately kept separate.
So in that case, Roger, your question, you would expect that you wouldn't get a difference because you'll get a robust estimate of the mean. But related to the previous question, I think if I understood Bill Slattery correctly, he said that the total error in the soil measurement was of the order of 9 to 22 tonnes per hectare under the strategies that he had available to him. what are you expecting your error to be, and what do you think an acceptable target is?
Ooh. Yeah, I am not sure at this stage. I think when you talk about, you know, errors of 22 tonnes and you look at my carbon distribution across that area that starts at 22 tonnes and goes up to 63, for accounting purposes, I think it's going to be quite different to what I'm trying to do within the carbon project. I guess, yeah, I'm going to have an awful lot of uncertainty associated with my management data in order to try and understand what's going on as well. So there's another level there as well.
It seems to me it's important to get on top of this issue, because it may well be that you can end up taking a lot of samples that don't actually give you any additional information in terms of error, because even under the strategy that you're using, you're not taking anything like enough samples to characterise the spatial variability. So there's a benefit cost issue that has to be addressed here in terms of the additional value that accrues from those additional samples, and I'm not all that persuaded so far that that benefit's necessarily accruing.
John did some work on some of his perennial sites where he looked at how many samples he would need in order to account for the variability within some of the perennial paddocks. But again, that is going to be very much dependent on the type of landscape that you're in and the amount of variability that you're dealing with. Which is pretty big issue to tackle, and it needs to be done.
Seems like we're getting a few questions, but I think we've got time for one more. So.
Mine's part of a comment actually, which relates to what Rob said and also what you've just been discussing in relation to sampling. And there's two issues. One is obviously the science of what we're doing and being able to be accurate and be able to predict. The other issue is in relation to carbon accounting and the cost of sampling. Mm-hmm. And to actually be accurate enough, it's going to cost you more than the carbon's worth half the time. So the question about how much sampling and how accurate will actually depend to a certain extent on the value of the carbon. So that really needs to be factored in terms of when you start talking about certainty and sampling strategy is, well, who's paying, and that'll be determined by carbon price.
Mm.
I think we'll wrap up that talk now, so thank you, Ben.
Thank you.
Speaker is MacDonaldSmernik from University of Adelaide. He's a senior lecturer in the School of Agriculture, Food and Wine, and he's been involved in soil organic carbon research over the past 14 years since starting as a postdoc in 1997.
Thanks, Matthew. Right, I'm actually quite familiar with this lecture theatre. This is where I give most of my lectures. Good to see a few faces who actually have attended those lectures. Hopefully they're not the disruptive ones. Okay, a little bit for those who don't know who I am, about me, some stuff about where I was born and where I've lived and what I did as education. My wife seems to think I overdo the country boy thing. As you can see, I was born in Griffith. My parents were, at the time, share farming my grandparents' farm. I like to think I was really involved, but my wife points out that I probably couldn't hold my head up by the time I moved to Sydney. So I'm actually a bit of a city slicker, and also in terms of agriculture, I came to it reasonably late in my career. My background is actually in chemistry and straight science, and it was only from my postdoc on that I've been involved here, but that is starting to be a fairly long time. So in summary, I'm a scientist, so my training is in chemistry and my research experience is in soil carbon generally. Here I worked initially with Malcolm Oates and since then kind of independently, but a lot with Jeff's group in CSIRO. Okay, second thing, what is biochar? And in terms of material science, it's a pretty straightforward question, and really it's just charcoal. I think most people know what charcoal is, but in case you don't, there's a bit, you can pass that around. As far as me as a chemist goes, it's just another name for charcoal. But in this context, in the soil carbon context, it has a bit more of a specific meaning. It's charcoal that's produced with the purpose of adding it to the soil. It's produced under conditions of pyrolysis of biomass rather than combustion of biomass, inefficient combustion of biomass. I'll go into that a little bit in a minute. And that biomass is generally waste biomass, either forestry waste, urban green waste, rice husks. There's a number of different types of waste biomass that can be utilised, and the other way is to specifically produce biomass crops and turn them into biochar. Heating temperatures we'll see in a minute that is quite important. Generally, the heating temperature for the biochar is around 400 to 600, but there's quite a bit of variability in how people are producing this stuff. Okay, that's what biochar is. The biochar concept sort of has these three prongs. The first is producing energy from the biomass. So by pyrolysing, by heating biomass in the absence of oxygen, it's exothermic, you produce energy from that, and it's one of the many ways that you can get energy from biomass. The second is that it has potential for storing carbon in soils, and the third is the potential for improving soil productivity. And I'll go into more detail of each of those as we go through. Okay, the first thing is, first part of it, actual pyrolysis, producing energy from biomass is the bit I know the least about, so I'm just parroting what I've heard from other people in the area. So basically what we're doing is heating biomass to around that 400 to 600 degrees Celsius without air or oxygen, and when you do that, you convert about 50% of the carbon into volatiles, into some CO2, but also gases and oils, which you can then combust with oxygen to produce energy. So that's sort of the energy component of it. You get some residual heat, which you can use to sustain that pyrolysis. So it's self-sustaining. You're not putting energy in to heat it up. Obviously, when you're starting the process up, you'll need to have a source of energy, but overall, the process of pyrolysis is self-sustaining, and you get a net energy output, and that's the energy side of it. There's some advantages in pyrolysis as a means for getting, if you like, bioenergy or energy from biomass. It's relatively low tech. It's efficient at fairly small scales, and this is important when you're talking about waste biomass, which is bulky and not particularly valuable, that if you can produce the energy on a small scale, you reduce transport costs. You can use most biomasses as a feedstock, not particularly picky, and the benefit in terms of greenhouse gases is that it's a replacement for fossil fuel, so we're getting energy directly from that biomass. And compared to other technologies for getting bioenergy, it's and efficient. I've got that in inverted commas because there's a lot of ways to define efficiency in these processes, but you're certainly converting a greater proportion of that carbon into something that you're using than if you're producing ethanol from sugar or corn or biomass, where you've got a fermentation procedure going on, and a lot of that energy that's stored in the biomass is actually being utilised by the bugs that are doing the transformation. Okay, the second part of it, which is probably the one you guys are most interested in, is storing carbon in soils. And the question is why we want to do this. I've given similar talks to grower groups and so on. And the answer, I go into a little bit more detail, but basically we're talking about greenhouse effect and global warming and all that kind of stuff. And I've assumed that other people have covered this already, or that we're already over the idea of why we'd want to store carbon in soils. Going into just a couple of specifics of storing carbon in soils. Why are we targeting soils? Well, they store a lot of carbon, more carbon than there is in vegetation and more than there is in the atmosphere, or actually more than the combination of those two. And the second part of it is that the soil carbon pool, these terrestrial pools, as compared to geological pools, which are really big, or oceanic pools, which are bigger than the terrestrial pools. The thing about the terrestrial pools is they're very strongly coupled to atmospheric CO2. About 16% of the CO2 in the atmosphere is pulled down out of the atmosphere by photosynthesis each year. But of course, nearly all of that is subsequently released through respiration by the plants themselves, by animals that eat those plants, by insects, bacteria, fungi, and so on. About 50% of that respiration happens above the ground, and about 50% of that respiration happens in the soil. So in the case of biochar, we already have the process by which the carbon is being taken out of the atmosphere. What happens to most plant material after it dies is it's broken down relatively quickly, and John has talked quite a bit about this in his talk. He also mentioned that charcoal, by contrast, is relatively inert or stable. It doesn't get broken down quickly by bacteria and fungi. And the idea of biochar, which if you do a Google image search for biochar on the web, you'll always come up with this particular diagram. What it's showing is that generally what's happening in the absence of utilising biochar, what we're doing by photosynthesis is we are fixing a lot of the carbon dioxide, but respiration is balancing that, respiration both by the plant and indirectly through the soil. The length of time that carbon that's been fixed by photosynthesis that is staying out of the atmosphere is fairly short, and we get released relatively quickly straight back to the atmosphere. The idea with biochar is to interrupt this process. So we're taking that 50%, say, of fixed carbon that isn't directly respired, that comes through in plant residues, pyrolysing half of that 50%, so 25% of the photosynthesised carbon is directly converted into carbon dioxide. We're getting some energy benefit out of that, but the remaining 50%, so 25% of total photosynthate, is then converted into a stable form. So by doing this, by converting 50% of the biomass to biochar, we're effectively shunting that 50% out of the rapidly cycling carbon pool. So key to the whole concept is this idea that biochar, is stable in soil, and first couple of speakers talked about this. This is something that's generally accepted, and there's sort of three reasons why we're pretty confident, or very confident, this is true. We've measured the rate directly of biochar carbon decomposition and compared that to the rate of breakdown of biomass. The second line of evidence is that we find a lot more charcoal in soil than we'd expect to find based on the rate at which it's added. So we're adding a lot of fresh plant residues. We're only adding, in natural systems, charcoal through fires. Fires are actually quite inefficient at producing charcoal. It varies with the type of fire and the type of vegetation and a lot of things, but generally when a fire goes through, only about 2 to 5% of that carbon will be converted into charcoal. Most of it, because it's burning in oxygen or burning in air, will be converted all the way through to carbon dioxide. The third line of evidence is carbon dating charcoal that we can obtain from the soil, either a charcoal fraction or pieces of charcoal. When we do that, we generally find it's older than the average soil carbon age. So, going a little bit more into these first couple of lines of evidence. So, measurements of the rate of decomposition of charcoal. Here's some work that Jeff Baldock and I published quite a few years ago now, where actually Jeff did this work, I did some other parts on the paper, but it gets cited a lot, so I'll claim it. And it actually gets cited because of this part of it, not because of the part I did. Anyway, comparing the rate of breakdown over 120 days of different types of carbon. Glucose breaks down very quickly. Cellulose, which is pretty much what most of fresh plant material is going to be, we get about 70% loss of that carbon to carbon dioxide, so 70% mineralisation over the 120-day incubation. When we heat that cellulose, or that wood in this case, we see a decrease in the amount that's respired over that 120 days when we get to materials that I'd call charcoal, which is really only right at the end there. There is very little decomposition at all. So these are the rates of decomposition of fresh biomass and charcoal are really chalk and cheese. They're different by orders of magnitude. Now, of course, it's going to depend on the conditions of incubation. A lab incubation is not going to be the same as what happens in the field, and there's all kinds of complications. But I think the main thing to recognise is that charcoal is inherently stable towards decomposition. Okay, the second line of evidence is the amount of charcoal that we find in soil, and fortunately the first two speakers actually spoke about this quite a bit. This work is based a lot on what was done here before my time, and when I first came here in the CSIRO group, particularly the work of Jan Skemstad on this, where he developed a method for quantifying charcoal in soils and showed that in a lot of situations, a lot of soils, there is quite a bit of charcoal that's there. This is a figure I pulled from one of those papers. It's actually quite a difficult thing to do, to measure this, but and there's been a lot more measurements than these, but just in general terms, it demonstrates the point that we can have charcoal representing up to 30% of carbon in a small number of soils. This is sort of the original work. To get it accepted around the place, he had to show that the same happened in Europe and the US, which he did subsequently in a couple of other papers. And this is sort of the basis from which the work on fractionation that John talked about comes from, and they'd have much better numbers on what the proportion is. The important thing to note, though, is that there's a lot more charcoal in soils. It represents quite a big proportion, a relatively big proportion of the total amount of carbon in soils compared to the amount of carbon additions to soils, which is very small in any given year. Okay, the third part of the biochar concept, if you like, is the effect of biochar on soil productivity. Now, we know that organic matter improves soil physical and chemical properties, and that's actually the reason for going to things like no-till. One of the reasons for going to no-till and developing methods, farming methods to increase the amount of carbon that's in soil, is that the organic matter is good for soil fertility and soil productivity. It's good for things like improving water holding capacity, rates of water infiltration, structural stability, and retention of nutrients, and providing a slow release form of nutrients. Organic matter does all these roles. What about biochar? Is biochar going to behave in the same way as other forms of organic matter? And the positive bit of it, or why we think that might be the case, at least in the long term, comes from the beneficial effects of char in what are called terra preta soils of the Amazon. So these are patches of dark soils that are found in the Amazon rainforest that are much more fertile than the surrounding soils. These soils are unusual for a number of reasons. They're very rich in organic matter, whereas, and this shows, compares the soil pits in a terra preta soil and an adjacent soil that is an oxisol, so a non-terra preta soil. So we have this dark colour associated with much higher amounts of organic matter. A lot of that organic matter is charcoal. Perhaps most of that organic matter is charcoal. The charcoal that's in those soils, we can measure we can carbon date and establish that it's very old. The other thing about these soils is that they are always associated with human occupation or evidence of human occupation. They appear to be sites where the native people have in the past cut down and burnt the rainforest residues, whether they were doing that on purpose or whether it's just pretty hard to burn forest in a wet climate, and so you end up with a lot of charcoal. Whatever the case, we end up with these soils that are very high in charcoal. And as you can see, the fertility of these soils is much greater than the fertility of the surrounding soil. So the simple conclusion you might come to from that is putting charcoal in soil is a good thing to do, and it's going to turn a weedy looking crop of corn that's a bit yellow and up to your knees into something that you're going to get lost in. This is, of course, the best case scenario. So why is this not going to be the case whenever we add charcoal to soil? And there's really two reasons for that. The first is that the aged charcoal, the charcoal that we find in these terra preta soils, is not the same as fresh charcoal that we produce. And the second is that not all soils and climates are like the Amazon. So, why should age matter for charcoal? One of the big benefits of aged charcoal in these soils seems to be the cation exchange capacity that they have. Cation exchange capacity is the negative charge on soil particles, and it allows soils to hold on to nutrients in a way that they don't leach, they don't get moved by water movement down through the profile, but they're not held so strongly that plants can't get at them. So the source of cation exchange capacity in the terra preta soils comes from very large negative charge that exists on aged charcoal particles. The point about fresh biochar is that it doesn't have this negative charge, and therefore it doesn't have a high cation exchange capacity. It has virtually no cation exchange capacity. So there's an important process that happens to charcoal over time. As charcoal gets oxidised, that oxidation of the char particles themselves creates a negative charge, and it's this negative charge that is responsible for the cation exchange capacity, the ability of these soils to hold on to nutrients against leaching. And going back to these NMR spectra, I'm usually when I give talks, I'm the one presenting the NMR spectra. You've already seen a few. I've tried to limit myself, but what I'd like to point out here, this is some NMR spectra of soil clay fractions and silt fractions with different lengths of photooxidation. This is a process that Jan developed as a method for isolating charcoal from soils, and what happens is as you oxidise the organic matter, the soil clay fraction, in the presence of oxygen and UV light, what happens is that the charcoal is resistant to that treatment. So as John said, this peak at 130 is the main one due to charcoal, and with photooxidation what we're getting is loss of all the other types of organic matter, and the residual is mostly charcoal. The important thing to note from here is this little peak here. This little peak is due to carboxyl groups on the charcoal, on the aged charcoal. We don't see that at all in fresh charcoal. And trying to quantify how much carboxyl, there can be up to maybe 1 in 10, 1 in 20 of the carbons in aged charcoal will actually have a negative charge, will be or potential negative charge. It'll be an acidic carboxyl group, and that is where the CEC comes from in aged charcoal. Okay, the second part about CEC, if CEC is why aged charcoal is useful, is the soil and the climate. So what's so special about the Amazon in this story? Well, we're talking areas that are high in rainfall, so leaching is a big problem. So if nutrients are in a soluble form, they will be, they have a great propensity to be lost, whereas in lower rainfall areas that's not such a big problem. Another point about the Amazon soils is they contain different types of clays to the types of clays that we have. Generally, they have clays that don't have much cation exchange capacity. In most soils, it's the clay minerals that contribute the cation exchange capacity or are responsible for the cation exchange capacity of soils. In the Amazon, a couple of things. One is that they tend to be acidic. In acidic situations, you have a lower cation exchange capacity. They also have clay minerals, and I'm just trying to catch the eye of Jock to say, check I'm not getting this wrong. They tend to have oxide, iron oxides, aluminium oxides, or kaolinite, which are the clays that have a lower CEC. So these two things together, the CEC of aged charcoal and the environment and soils of the Amazon, means that if there's anywhere that aged biochar would be a good thing, it's going to be in the Amazon. So I guess what I'm saying is that expecting differences with addition of biochar like that in terms of productivity is probably being very, very hopeful. However, there's certainly some recent research that has discovered or identified fertile char-rich soils elsewhere, particularly in Europe and Australia. So it's not something that's specific to the Amazon, but certainly that's the first place it was recognised, and it's certainly where you'd expect that phenomenon to happen. I said one of the examples is an Australian example of aged biochar providing a very different soil to the surrounding soils. This is some work I did with a number of other people, and it's been recently published on what we call Terra preta australis, so these terra preta soils in Australia. And these soils have been developed on areas where charcoal deposition over many centuries has happened. These are Aboriginal oven mounds sites along the Murray River, and The thing to note is, again, we're seeing this difference in the colour of the soil, so we're getting very dark soils where we have a lot of charcoal, and the amount of carbon that is stored in those soils, whereas the adjacent soils, we see the normal profile of soil carbon, so starting, in this case, around 3-4% and quickly dropping at 10 centimetres to 1% or less. These Terra Preta australis soils have much higher carbon contents all the way through, and we've shown that a lot of that is due to the presence of char. And once again, even though these soils aren't as low as in CEC as the rainforest soils in the Amazon, the presence of that aged biochar is making a big difference in the cation exchange capacity. Now, in terms of how long does it take for this process to happen, well, in the end, what we're going to have to do is add biochar to soil, or add chars to soil and see what happens. So the ultimate way to see if biochar is going to provide a fertility benefit is to add it to soil and grow something on it. And the last talk this afternoon, last talk before the tea break, I think it is, I'm just having a look at the program. No, after the tea break, Greg Butler is going to talk about a number of field trials on soil carbon, but some of those are going to include biochar later on, so I won't go on about those. There has been a lot of field trials initiated, but interpreting the results of those is not entirely straightforward. Some reasons for this, if you look in the literature, of course you're going to be more likely to see examples where it's been successful because it's easier to publish stuff when things are successful. If it does nothing, then it's awfully hard to get excited about it and to be bothered sitting down and writing it, and it's hard to get it published. The initial addition of biochar isn't just the addition of carbon in the form of the aromatic structures. There's other stuff that is going to be there, in particular ash, and ash contains lime. It'll often contain nutrients, particularly phosphorus. So where you do see an effect of biochar, it doesn't necessarily mean it's the carbon that's doing something. It's not necessarily that Terra Preta phenomenon. It might be more about the addition of the ash. Another difficulty in field trials involving biochar is that where do you start? You can't cover all possibilities. We have biochars produced from different feedstocks at different temperatures, different heating rates, and then there's soil properties, so different soil types, different climates, different crop types, different managements, and so on and so on and so on. So as we get further into the field trials and start to see some of this complexity, what we're finding is that it works in some places, doesn't work in other places, works with some chars, doesn't work with other chars, and so on. So rather than going about it randomly, I think it's probably, at this stage anyway, probably better to think about why it might work, and it's only going to have an effect, a beneficial effect, if on productivity, if it limits some limiting factor to crop growth. So if there's problems with water holding capacity and it provides water holding capacity, then that is possibly going to improve things. If soil strength is limiting, the addition of biochar may decrease soil strength, and in that case you might see an effect. Also, things like water movement, pH, the effect of the ash component may be responsible for an effective addition of biochar if there are nutrient limitations, or if it has an effect on the biotic factors of soils, if it has some effect on disease or beneficial organisms. So I think that the point here is, with biochar field trials, is to try and understand the mechanisms where there is an effect, or where there isn't an effect, not just whether it works or it doesn't work. Okay, I was in a previous talk I was asked whether biochar was a myth or a miracle, and this is my My feeling, it's neither. It's not a myth, in that pyrolysis does provide an effective way to convert biomass into energy, and that potentially is a way to go to deal with energy issues. Biochar is certainly stable in soil, so converting biomass to biochar will certainly result in an increase in carbon stored in the soil. And there's no doubt that aged charcoal in soil does provide a fertility benefit in some soils, and we can see that directly. However, it's not a miracle. People have been using biomass as an energy source since before they used fossil fuels, and the reason why we went from that is because it has disadvantages. And the production of biochar simply to provide a stabilised form of carbon is not economic at this point, and probably never will be. And in terms of the effect of biochar on productivity, you shouldn't expect to just chuck some charcoal on your land and double your harvest. It ain't going to happen. If it did happen like that, I think someone would have worked that out. It's not as if we haven't been farming and haven't had charcoal and haven't put the two together at some stage. So, in my opinion, the whole idea of biochar has a small but not minuscule chance of being really useful in this area in terms of carbon sequestration and effects mitigating climate change, and probably a moderate chance of being fairly useful. The upside of biochar, we know that organic matter is useful to soil fertility, and we've changed the way we farm in recent years in response to that. We are likely to be able to tailor biochar properties so it provides an immediate benefit to produce a pre-aged biochar, if you like. And one of the reasons why we haven't seen the benefits immediately is that it takes this process of aging to get that cation exchange capacity. So there's certainly possibilities in this area. And in terms of carbon sequestration, all we're really doing is utilising photosynthesis, which is a very efficient and convenient way of getting carbon out of the atmosphere, and as I said, shunting some of that carbon that we fix through biosynthesis in a way that is going to keep it in the soil. So, downside, to make a real difference, we need to source a hell of a lot of biomass, and once we get away from sources of waste biomass, there's issues of competition with food production and water, and also the use and the fate of nutrients. One thing that happens during pyrolysis is you probably lose most of your nitrogen, and the rest of the nitrogen may be fixed into a form that is not plant available. So that is certainly an instance where you're losing something from against the alternative of adding that biomass or leaving that biomass on the soil. We probably need all three aspects of biochar to work: the energy production, the carbon sequestration, and the fertility benefit to make the whole thing economic. And of course, things always end up a lot more complicated once you start doing them. Getting the balance of all three of these prongs and optimising them against one another and finding the situations where it's most likely to work is going to be quite a long process. Okay, I think I've got a few minutes still. Couple? Okay. Little bit of where the research is at. So this is, like I say, I'm a researcher, and so this is from my perspective. There's currently two quite large collaborative biochar research projects funded by GRDC and DAF, and Evelyn Kroll in Cairo is running those from here on the Wake campus. There's a number of different aspects of those projects. One is the characterisation of biochars. There's trying to measure the stability of biochar, effects on crop yield and quality, life cycle analysis, effect on soil microbes, and interactions with pesticides, herbicides and contaminants. I'm only going to talk about the first couple of those. First one being biochar characterisation. What we found by looking at these large number of biochars that have been produced in labs or produced commercially is that there's huge variability depending on the starting material, the treatment temperature, so what temperature you're pyrolysing at, and the pyrolysis conditions. So in terms of starting material, it's going to affect the concentration of nutrients, the liming value, and also it has an effect on how charry the char is. One thing, once you get into charcoal chemistry, is that it's not one material, and there's a range of materials. Mostly, for any one material, how stable it is seems to depend on the heat treatment temperature, but if you start with a different material, you can end up with, at the same temperature, something that's more degradable or less degradable. And I won't go into those bottom two plots. In terms of biochar stability, this is a really hard thing to determine. How long is it going to last for? This is what we really want to know, but it's hard to measure or estimate something that happens very slowly. Now, the best data we probably have at the moment is from controlled lab incubations, measuring CO2 release from charcoal, not over a 120-day period, but over much longer periods. At the moment, the best data we've got is for 4 years of incubations. What we find is that we get an initial loss of carbon that's reasonably quick, slower than for biomass, but still sort of over that weeks and months period, and then it tails off. After 4 years, we get mineralisation of carbon between about 0.5% and 8%, depending on the production temperature and the starting material. And this converts to half-lives of anything between 70 and 1,000 years, but the meaning of a half-life, when you've only looked at the first bit of the incubation, you don't really know whether there's different components, is a little bit problematic. Another complication is priming effects. A priming effect is when you add something, one form of carbon, does it increase the rate at which the other organic matter decomposes or decrease it. And we actually find both positive and negative priming effects at different times for different charts. So quite a complicated thing happening there. And of course, even when we do these measurements over a long period of time, that just tells you how stable it is under those conditions. And it could be affected by other things that happen. Okay, this is my last slide, I think. So the effects on crop yield and quantity, these, and Greg's going to give a lot more data, I think, some other examples later this afternoon. They're very dependent on soil type and biochar type, and I think a lot of the immediate effects are probably due to liming and or nutrient effects, and there's a lot more that needs to be done. In terms of ASPAC issues about analysing, how will existing analytical methods see biochar? Well, it should be fully detected by dry combustion, although chars produced at high temperatures are quite thermally stable and they may be underestimated, so it's something that you probably should check. But in general terms, biochar will be detected by dry combustion pretty much as organic carbon. Walkley black probably will underestimate biochar, so you're probably not going to detect it at all. Whereas modified wet oxidation, things like the Heen's method, probably will detect most of the biochar, but again, it's going to depend on what that biochar actually is. Another question is whether analytical methods will be able to differentiate biochar from other forms of organic carbon. This is something that, as John said, is causing quite a few problems. For when we're talking about charcoal that's already in soils, it's not an easy thing to do, and there's certainly no existing absolutely reliable technique as yet. So, concluding, biochar concept is the conversion of biomass to bioenergy by pyrolysis. When we add it to soil, that biochar is going to be a lot more resistant to mineralisation than biomass, and that provides a potential for carbon sequestration. It's likely to also influence soil properties, and hopefully that's in a beneficial way and improve productivity. It's certainly an area of active research. There's a lot of unknowns remaining, and if you do want some more information, that's the Australian-New Zealand Biochar Research Network or something like that, and there's plenty of information on that. Thanks.
Well, we had a lot of... any questions?
Temperament.
So I'll just put in the voice. Ron, there's obviously been a lot of excitement around biochar in the past decade, and it's only gone up past some of the carbon farming initiative, but also the clean energy plan. It's interesting to see where the science is coming together nicely, but also the economics are in considerations to what biochar application is going to take off. If you were to have a punt, what type of application do you think we'll see really take off the next 5 to 10 years? And I suppose I've got in mind here options of mobile pyrolysis units going throughout the valley or pyrolysis on large-scale wood harvesting. What do you think might take a run?
I think probably the first cab off the rank will be waste organics, where there's biomass that has a negative value. People are trying to get rid of it, and certainly that helps if someone's willing to give you stuff for free or to pay for it. I guess, because it's got those three components, it's wherever one of those is particularly useful, it's more likely to be useful. I was listening to Radio National on my way to McDonald's for lunch yesterday, and they were talking about a situation in Cobar where they were looking at using the woody weeds that invade that area as a source of biomass, and also they're at the edge of the power grid. There's mining activities there where they need energy, so that energy part of it and a large and cheap or free or something that someone would pay to get rid of source of biomass is there. So I think it's probably in those areas where things are going to happen fairly quickly. I think the one that gets talked about a lot is crop residues, and I don't think that's really likely to be a place where it's going to happen quickly, because we've spent all this time actually breeding our crops not to have residues, not to have much in the way of chaff, and the residues that we have are important in nutrient cycling. They're important in holding soil together through the summer against water erosion, wind erosion. So I think we're unlikely to start gathering up that material and turning it into biochar. So I think it's areas where there's a source of cheap biomass that doesn't have another purpose, and where power is something that has a higher value than in the grid as a whole.
Ron, you mentioned that the cation exchange capacity of biochar increases with its age. Do you have any feeling of, so if you applied a reasonable amount of biochar, fresh biochar to like a red-brown earth in the mid-north, do you have any feeling for how long it would take before you had a significant impact on cation exchange capacity in that sort of soil and climate?
Yeah, well, I think I could narrow it down between 10 years and 1,000. If that's useful. There's certainly a lot of work in that area. it doesn't happen instantaneously, and that's probably why people aren't seeing a benefit, and that's why we haven't stumbled upon it before. It's going to depend on the climate and the soil as well. Johannes Lehmann has done some work in Kenya where does seem to be oxidised groups on the charcoal building up on that decade scale, but probably we're talking a higher temperature and higher rainfall. It's something that the ageing of biochar certainly happens more quickly at higher temperatures, but whether or not that requires the char to be wet, whether it's biotic or abiotic, probably a bit of both, and understanding some of those will take a bit of time. I don't think it's going to happen to any great extent more quickly than over a period of 10 years, and then the economic value of that becomes quite questionable. Like, who's going to do something that's going to have a benefit for down the track in straight economic terms?
Okay, question out there.
Oh, really?
I've already down to one microphone, so I'm slowing up a bit. Go ahead.
Really, the same question. I just wondered whether anybody had followed the oxidation using solid state C13 and NMR to see if anything, once you've lost that 0.5 to 8%, which obviously could lead to misunderstandings. I mean, over the next few years, and people have been going now for 10 years, has anybody tried to follow that oxidation?
Yeah, I have.
And
after the samples I had after 2 years, we don't see anything. Another situation was one of my PhD students who's here, Anna, has done some experiments on chars from bushfire sites. So we went up into the national parks in the Adelaide Hills, and where we knew when the most recent bushfire was, collected charcoal from those areas and looked specifically for a build-up of oxidised carbon there. We didn't see anything out to about 30 or 40 years, but it's quite difficult because the char that you can find in those sites tends to be large chunks that is coming from woody material from the larger trees. It's probably the finer material that's been incorporated in the soil that's going to build up those oxidised functional groups a lot more quickly. So, yeah, I did do some analyses on Johannes Lehmann's Kenyan charcoal soils. They were sites that had been burnt, purposely burnt, and they had some idea of when that had happened, and they ranged from, within the last year to 1 year, I think sort of something like 4 or 5 years, 10 years, out to 9 years. And certainly for the older chars in that series, we seem to be seeing carboxyl groups on the charcoal. Technically, it's quite a difficult thing to do, and there's other things that happen. For example, the biochar will sorb other organic matter to it, some of which has carboxyl groups on it. So just because you're finding carboxyl groups in your char sample doesn't necessarily mean it's coming from the charcoal itself. But whenever we look at the charcoal fraction, the resistant organic carbon fraction in soils, it always has those oxidised groups. So it happens all over the place, sometime between a decade and a couple hundred years. But yeah, we continue to work on it and to find situations where we have charcoal of a known age that's been in the environment, and we can quantify those processes.
We've got time for one more person down in front.
Thanks, Ron, for a really interesting presentation. Interesting talking. It needs at least high temperature for pyrolysis. And how will it produce this temperature, this mass temperature? it is nested with carbon dioxide production, or no? If you produce this much temperature by burning coal, so it produces some carbon dioxide, and it goes to a temperature, and also carbon dioxide is really a constituent of greenhouse gases. So what is the comments about?
Could you just repeat that?
So how do you get started?
How do you start the pyrolysis?
Yeah, it needs high temperature.
Yep.
How they create this much temperature?
Yeah. The pyrolysis side of things is not something I know a lot about. I think once it's going, it's self-sustaining because you get a lot of energy, and it's certainly an area of the engineering of pyrolysis units for this purpose is something that's continuing and improving. I think the idea is to get these things running at a constant rate so that the waste heat that you produce from actually burning the volatile materials, so you get energy from that, but there's waste heat that goes back into preheating up the material. But to get the initial starting, you'd have to use natural gas or something. There'd be an alternative source of energy that would get it up to a certain heat, at which time the volatile component starts to come off, and once you get enough of that volatile material, then you can circulate that back in towards the heating process.
But initially, we start with like charcoal or gas like this.
Yes. Yeah.
It produces some carbon dioxide.
Yes. And you have to take that into account. Yeah.
Just my question is actually, what is your comments about this? How will minimize this carbon dioxide?
I think by continuing, I think it's turning it off and turning it on is going to be inefficient. So it's about having a continual source of biomass that you're continually processing. But yeah, the processing side of things and is a different area. And I do know there's continual improvements in pyrolysis units for this purpose, and they can be tailored. There's trade-offs between the amount of carbon that gets fixed and the amount of carbon that goes to energy production. There's also, with temperature, there's also trade-offs in the nature of that charcoal. So you might want a lower temperature charcoal that ages more quickly and produces that productivity benefit. That seems to be the way things are going, or a higher temperature charcoal which is more stable and you get lower losses over time. So those kind of things can still be tailored. The hard thing is knowing how good your product is going to take a long time.